Welcome everyone. I'm very happy to have Ashesh with us today. He is a PhD student at Dresden, Germany, and he works in a lot of different places, more in terms of geographical places and also in terms of research places. I'm excited about this talk because this already got the award of the best oral presentation at the HP symposium. So let's have him.
Thank you very much for organizing this talk. I'd also like to thank Professor Kavita for giving me this opportunity. So, hello everyone. Today I will present for you my PhD work, which is an image decomposition task applied on fluorescence microscopy data. But let me start by introducing myself. So, I was born and brought up in Bihar, India. I did my bachelor's and master's in computer science from IT Delhi. And I moved to industry. I worked for 3 years as a data scientist in Bangalore. From there, I went to NTU Taiwan, where I worked as a research assistant in a computer vision lab, worked on different vision projects. From there, when I was there, I got selected for doing my PhD at Max Planck, Germany. But my professor moved to human Technopole, Milan, arguably because his wife is Italian. And so I have done all my research at human Technopole, Italy. So, yeah. So my idea is that I will try to explain to you what is a fluorescence microscopy, and from there we will move into the computer science aspect of this presentation. So let's ask this simple question: how do people image nucleus in fluorescence microscopy? So what they do is they attach some light-emitting compounds known as fluorophores to nucleus, and the property of this compound is that when you shine light of a specific wavelength, it gets excited and it starts to emit light. So what I mean is that you attach your fluorophore to the nucleus, you put your specimen under the microscope and you shine light. And what happens is it starts to emit specific wavelengths, and then you collect, and from optics you get an image. So that's how it works. If you want to image multiple structures, and please feel free to interrupt me at any point should you feel the need. So for multicolor imaging, what you have is you have two different fluorophores. You attach one of them to one structure type and the other to the other. And these fluorophores are specific in the sense that if you want to attach to nucleus, it will only attach to nucleus and nothing else. And in this way, in this case, you have two lasers. You shine light for one laser, you get one image. You shine light with the other laser, you get the other image. Now, what is important to note here is that it will only work when these two different fluorophores, they emit different wavelengths. Because from optics, there is a way to separate these photons based on the wavelengths. But if they all emit the same wavelength, then it will be impossible to segregate these things. Yes?
Several of us are from CS theory. So what's the fluorophore and what's the property of, what do you mean by when you throw light on it, what happens? And
okay, so I would be brief in my answer.
Yeah.
So fluorophore is a chemical compound, is a class of chemical compounds which has the property that when you shine light of specific wavelengths, they get excited and they emit photons of specific wavelengths. Okay. And so here what I was trying.
Fluorophore is associated with a particular band.
Exactly. And so you see, like this fluorophore will emit between 400 nanometers to 500, and this will from 300 to 400 nanometers. And so from optics, you can then segregate these two streams of photons into the two images. And so in this way, you get two different images.
And these occur naturally in the object that we are trying to...
No, you attach it.
Yeah,
you stain it. Yes.
These are
attachments. Yes.
Okay.
Yes.
If you don't have them, you won't be able to see the object.
With fluorescence microscopy, no. Yeah, you need them. You need them because you need the light coming from that specific structure that you'd want to image.
But if you just have like a standard, let's say some tube light, then would you see everything?
Yeah, so that is, so there is transmitted light microscopy, for which you do not need this, but then it loses the specificity. Then you get everything that is present in the cell. And sometimes you would just want to track some specific suborganelle. And so for that, this is used. Okay. And this brings me to the limitation. The limitation is that their emission spectra typically often overlap for the different fluorophores. So here I show the emission spectra of one of the fluorophores. On the x-axis, you have the wavelength. On the y-axis, you have the probability density. So this tells you what is the probability that light, a photon of this wavelength, can get emitted from that fluorophore. So people do not collect all this light. What they do is they have this emission filter which limits what is collected. And the reason is there are different light sources inside a microscope. And so if you collect everything, then the contrast of an image is poor. And so you need some sort of a filtering mechanism to filter out the unnecessary light coming from other sources. The problem is when you have two fluorophores, then their emission spectra overlap. And so now you need a much narrower emission filter, which means that you're not collecting all the light that is emitted by those specific photons. And so what you get is a low SNR image, a noisy acquisition, all bad things, essentially. And so let me walk you through a set of, let's say, decisions that a typical microscopist makes. And that will, let's say, will help you understand the limitations of this modality. So let's say we always want to collect more light. So one option is, why don't we have a broader emission side filter? If you have it, what you will have is you will also collect the light that is coming from a different fluorophore, this one. And so the acquired image will contain your structure, but it will also contain the structure that was present in the other fluorophore. So what you will get is a superposition of these two structures, which is something that you don't want. So this is called bleed through. And so you don't typically do this. So the downside in not doing this is that you have low light collection efficiency, which means noisy acquisition. One way to improve it would be to increase the exposure time. That is, you collect photons for a longer time. And if you do that, then you are good, but the speed is less. If you want to image, let's say live sampling, if you want to do live imaging, then you may want to inspect your sample at a higher frequency. And so you cannot do this if you use multiple fluorophores. The other option is you can increase the laser power. That is a possibility, but that has other issues like phototoxicity, photobleaching. Essentially, you don't want to bombard your specimen with too much light. So that is also a negative thing that people don't do. This brings me to the limitations of this modality and the ones that we want to address. First is you cannot use too many.
I have a question.
Yes.
Suppose you do this many different times with lights from, let's say, different angles, and
But cells are moving, right? So you cannot capture the same thing.
let's say you do it simultaneously with lights from different angles, and you take let's say the majority of the different fluorophores. Would that be more accurate than one?
It would be, but the problem is that with a single fluorophore itself, the cells are almost burning in the sense that they don't like too much light. And if what you're saying is that I will bombard it from 10 different ways, right? And you could do it, but I'm sure people would have tried it, and due to the reasons that I just mentioned, it is not used.
Okay.
Yeah. So the main limitation that my PhD caters to is the first one, which is you cannot use too many fluorophores concurrently, because when you use multiple fluorophores, your collection efficiency reduces. And so what this means is you cannot image multiple structures concurrently. So in practice, people do not image more than 4 to 5 different structures. And this is something that my work tries to enhance. The second is we already saw that you have the light collection efficiency, which either makes it slow or makes it noisy. Yeah. Now let's go to our approach. So the aim is to improve how multicolor imaging is done, and we want to image more structures, we want to image faster, and we want to acquire less data and handle the noise. We want to acquire less noisy data and handle the noise in case the noise is there. And what we are saying is that we want to image multiple structures into a single channel. So that is kind of the idea. So what you do is, if you have 2 fluorophores, you shine both lasers together. So the photons from both fluorophores are excited, and you have a broad enough emission side filter which will collect all the photons. So what you will get is a superposition. You will get a superimposed image. It solves the low light collection efficiency because your emission side filter is broad. But the problem is you do not have two structures. You have a single structure now, which is a superposition of these two things. And now the task is, can we go from here to the individual structures? So it makes it faster. The other way is that you can use a single fluorophore to image them together. Again, very similar. It is just that we will just use a single fluorophore and not two different fluorophores. And in this case, you are using just one fluorophore to image two things. So you could image other things in the other channels that are now freed. So this is the task for my PhD. Given a superimposed image, the task is, can we get individual constituents? And so for this, the network somehow needs to detect the two structures that are present in the input, and then it needs to say, okay, this structure needs to go here, and that structure needs to go there. And for this, early in my PhD, we observed that spatial context is needed to detect those different structures. For example, if I ask you, is this image an image of belongs to F or E, you would not be able to say. But if we give slightly more context, still not possible. More context, still not possible. But when you have sufficient context, then you could say that, yes, this indeed belongs to one of the channels. And in hindsight, so it was not surprising that we found that larger patch sizes improved the performance when we trained the network on this task. But the problem here is that larger patch sizes means larger GPU consumption, and most microscopy labs cannot afford it. And so this was our first work, µSplit, where we tried to incorporate this notion of spatial context in a GPU efficient way to solve this task of image decomposition. So I will, let's say, try to explain this idea by an example. So let's say you have this.
So the question now is image decomposition is, you're taking all the image and now it's a software problem of trying to decompose the things.
Right. So from microscope, we will get a superimposed image, and from there we want to split it apart, right? So the first two works that I will present, the input will be simply a sum of the two structures. Later, I will show you results where we have actually imaged structures, superimposed structures directly from microscope. So let's say this is your frame, big frame, and you would want to predict for this small rectangular patch here. So what we do is, so this is our primary input for we want the two predictions. What we do is we take a larger patch which is centered on the primary patch, which looks at a larger region, and then we take more, and then we take more. And the idea is that we want to feed all of this into the network, and to get from the network the two predictions corresponding to this primary patch. And I hope you see that with these extra inputs, we are able to give larger spatial context. But if you were to do this naively, then it will take more GPU resources. And so what we do is we downsample all these extra LC inputs to the same, let's say, pixel dimensions as the primary input. So if the primary input is 64 plus 64, all the LC inputs are also of the same size, 64 plus 64. And it makes intuitive sense, because if you want to predict for one patch, then the further away you look from that patch, you don't really need the high frequency details. You could do away with high frequency details. And as long as you have, let's say, a broader idea of how this primary input fits in the larger scheme, that is good enough. We train this setup in a supervised way, in the sense that we have access to input, we have access to the two channels, and we train the network. Now that we have some idea of how we are trying to solve this problem, let me go into a bit more details. So this architecture is actually a meta-architecture, in the sense that we will integrate this with UNets and hierarchical variational autoencoders. The requirement for µSplit is the architecture should have an encoder-decoder based setup, where there is this notion of hierarchy levels, where at every next hierarchy level, the spatial size goes down by a factor of 2 in the embeddings. And you can ask why that is the case, because we also have a notion of hierarchy levels here in our inputs, because the first LC input is downsampled by a factor of 2, the second is downsampled by a factor of 4, third by a factor of 8, and so on. And so it made intuitive sense that we merge the information of, let's say, the first LC input with the first hierarchy level, the second with the second, and third with the third. And so that is what we did. And for doing this, we just zero padded the primary embedding so that you have the same spatial dimensions. And then we concatenate the embedding coming from the respective LC input. And this is our first variant, which we call as lean LC. So we provide 3 different variants for µSplit. They all differ in how much GPU resources they consume and how much performant they are.
So this autoencoder is already trained or you train it?
We train it, yes. Yes.
So the input is actually a image, like is it a slice or a stack? And is it?
At this point, it is a slice. It is a slice. 2D image, yes.
Okay,
Right.
and it is basically, let's assume that 2 different structures stained with the same dye.
Yes. Or 2 fluorophores, but you have both of them turned on simultaneously. Yes. So here, my claim is that only encoder actually benefits explicitly from the context, because this is the gray region which I represent. This is the extra portion that was added because of these LC inputs. And when we pass this information via skip connections into the decoder, we center crop these extra regions. The other variant was regular LC, where we do not do this center cropping, and so both encoder and decoder benefits explicitly from the context. In here, I would just like to mention one of the limitations of units, which is that they cannot be, let's say, they have a limitation in how much deep you can go. For example, if you start from 64 cross 64, then you could go to 32 cross 32, 16 cross 16, 8 cross 8, 4 cross 4, 2 cross 2, and 1 cross 1. You cannot go deeper than that. But here, since the spatial dimensions do not change, we had the option of stacking few extra layers in between, and which is what we did. And so deep LC is essentially regular LC plus more depth in more number of hierarchy levels. So now that I have explained to you the different variants, let's now move to the architectures. So we integrated µSplit with UNet and with HVAE. With UNet, it was relatively simple, no issues. But with HVAE, we had to do several changes. So let me just briefly introduce what is a variational autoencoder. So this is an encoder-decoder based architecture where the encoder does not give you an embedding directly. It gives you parameters of a distribution, and then you sample from that distribution to get the embedding. And then you feed that embedding into the decoder. And this task is typically reconstruction. So you reconstruct the same thing. Hierarchical VAE is essentially a VAE which has multiple hierarchy levels. The same things that we saw before, different hierarchy levels. And we needed to do many changes to make µSplit work with the HVAE. So I will briefly go through each one of them. So the first part was we needed to make sure that we are theoretically sound. So because our setup is not an autoencoding setup, the input is a single channel and the output are 2 channels. In a VAE, you reconstruct. So input is 1 channel, output is the same thing. So in a VAE, you start by maximizing the log likelihood of the data that you have, and you introduce this notion of latent space. You apply Jensen's inequality, and you end up with this 2-term expression that the first term is log likelihood, and the second is the KL divergence. And so we followed the identical steps, but we started by a different objective. We wanted to maximize the joint log likelihood of the 2 structures.
So what is the training data here? What are you training the autoencoder on?
So now it is not an autoencoder because you have 1 channel input and 2 channel outputs, right? And the input is a superposition of 2 structures. Those 2 structures are the 2 targets.
So you have some label data where you already know what the 2 things are.
Yes.
So you have where you get you probably need a lot of that for training, right? A lot of data in this format. So how do you get that?
So I mean, we sit in a biological institute, so we have access to microscopes. Yes. So yeah, so it is a supervised task, if that is what you wanted to ask. Yes.
So that question related was Hari's question on the previous question. So what exactly is the input to the problem, the image taken or even this whole the corpus of training data? For example, let's go back to the example which you said, the E and the F. So you had an image, you had a small subpart of an image.
Right.
And
yes,
then you knew the corpus of data was all rendering of the 26 alphabets, so possibly you were able to do this. Similarly, what you have is the corpus on which you're training to.
So we have this big frame. This is 2000 cross 2000. This is our input. This is a superposition of two structures. And along with this frame, we also have the individual constituents, two channels, two targets. Okay. And since this is large, you cannot train it on the full image. What you do is you take small 64 cross 64 size patches. So now you have 164 cross 64 sized patch, which will be, let's say, a superposition of letters A and B. And then you have two corresponding patches, which will contain parts of A and parts of B. And then you train the network to go from this superposition to the two structures.
I'm still confused. In the case when you had E versus F,
right?
Do you have this prior knowledge that the two components are alphabets?
Right. Here also we know that one of the channels will be nucleus and one of the channels will be tubulin, some structure. So we have this knowledge that the first channel will only be one specific structure and the second channel will always be the other structure.
I see.
So it is not that we are trying to disentangle, let's say, in unsupervised, because then you could split arbitrary structure types. This is not what we are doing. We know that we are working to split nucleus from tubulin or some other structure, and we have the data, paired data for it.
So you have a specific type of cell, you know what the nucleus looks like, you know what the tubular looks like.
Yes.
This one is separate.
Yes.
This is more like object identification there.
Identification, you could say that, but it's just that we will not have. So if you are talking about bounding boxes, then you will not have the structures, right? Because they are superimposed. So when we look at the results, I guess you will better appreciate that it is difficult to identify things.
But there are also these works on foreground, background separation, things like that. How related are they?
They are different because foreground, background means that you in the background essentially there is nothing, right? You could threshold, that's what you're saying. But here you have two actively present structures, they are overlapping. And so, for example, when I don't have the results for this image, but essentially there are two structures overlapped here. So it is not trivial to separate them through, let's say, classical measures. Yes.
So your training data is acquired using the classical ways on focal ways.
Yes.
Narrow.
Yes.
And just for my clarification, if I would have missed that, you are using the data which has used only single fluorophore or multicolor?
Multicolor.
Okay. So in the context of the emission spectra, how is this going to help resolve the superimposition in the...
So you will get a superimposed image, right?
Okay.
You will get it. And then we will use deep learning to split the superimposed image into individual constituents. So the benefit is that now you don't have a narrow filter. Now you have a broad enough filter, so you could collect more light.
So maybe you will come to this, but this will work fine maybe for the physically segregated organisms or whatever constitutes in the cell, within a cell. But imagine if it's an overlapping sequence. Would this also work? You will come to
Yes. I have good results. That is what I think. Yes. Any more questions? Yeah, so yeah, this part was done. The second part was if you have trained variation autoencoders, deep variation autoencoders, then you know that they are unstable when you train such networks. And so one of the reasons that I found of their instability that you typically have to take this exponential function on the output of intermediate things. So encoder's output is mu and log var. And from log var, if you want to go to variance, you would take exponential on this thing. And so if during training you get a large enough value, then taking the exponential breaks everything and things becomes unstable. We looked up the literature and we found one of the ways that people worked was they model the variance in a different way. So now if the network's output is less than 0, you take variance as exponential of x. If the network's output is positive, you model variance as 1 plus x, and so you avoid applying exponential on positive values. This stabilized 2 of our variants, but when we tried to train DeepLC, it was still not working. And we again looked into the literature and we found that that people typically divide their posterior parameters, intermediate activations by some factor. And so
you're saying that this function of e to the minus x, e to the x and 1 plus x, it works better than sigmoid or ReLU or something?
No, but sigmoid has a limitation, right? You cannot go above 1. And
Yeah.
here we would want to go above 1. Certainly, we just don't want exponential of 20. That will be huge. So we just want to limit how fast it grows.
But it could be 1000 unit here.
1000 is fine, yes. So the problem is when you have exponential raised to 20, for example, then all the gradient computations, everything breaks. So even if you have 1000, there will be a spike, but the network will be able to come back to where it wants to be. But when you have very large spikes, then you cannot go back. Yeah, so we scaled our let's say intermediate layers by this factor. Don't ask me why did we use square root of 2n here. N is the number of hierarchy levels. Different papers use different factors to scale down their parameters, and we just picked one of them. The last part was on KL loss reweighting. So this was to improve performance. Others were just to make things stable. So in the VAE, one of the most, let's say, popular features of a VAE is that it gives you denoised images. And the way it does is, it does not allow the information to pass from encoder to decoder. It does not allow to pass the high frequency details from encoder to the decoder. And this is achieved by this scale divergence loss. In our case, we were working with high resolution data. And so we wanted these fine grained details. And so we weighted the KL divergence in such a way that the high frequency information was allowed to pass from encoder to decoder in a better way, and that gave us better performance. Now, we did so much for that to make HVAE work. Now, the question that one can ask is so much effort really needed? So the question is, does variational nature gives you any performance boost? And so we created a different architecture where we took HVAE and we removed the variational nature from it. So essentially, we removed the stochasticity. So all the other parameters are identical. And we found that indeed the performance was better. There are other, let's say, use cases of the variation nature that I will show in the second part of my presentation. Let's now look at some numbers. Here we have 4 tasks: actin versus nucleus, tubulin versus nucleus. These are different, let's say, cellular components, the different structures. Actin versus tubulin and actin versus mitochondria. I show you the PSNR and SSIM values. The larger, the better. The second column is the GPU consumed during training. I want you to just focus on 2 parts. The first is we trained a unit with a patch size of 512, and it took quite a lot of GPU resources for training, and we were still able to outperform it using our DeepLC, which took less GPU consumption, and it was trained with a patch size of just 64. Of course, it should not come as a surprise because the additional LC inputs indeed capture the context. So you really don't need large patches. You could just train with small patches as long as your primary input is in the full resolution. And the second part, if you compare the lean LC, regular LC, and deep LC, then you can see that in terms of GPU, they consume different amounts of GPU resources.
It seems marginal 0.79, 0.81, how significant is that?
Which ones? The SSIM values don't change a lot, so
Even the other values, 33.3, 33.9.
so are you comparing our methods or are you comparing with the vanilla
one
ones?
with the green, the old one with the new.
The old one. So you mean this or this one, which is
The red rectangle.
yeah, but this is trained with a patch size of 512, right? And this people cannot train. That is essentially the thing that we cannot. People, microscopists cannot use this architecture. And what we are saying is that we are doing even better than them. So this is something that people cannot use. What they can use is the vanilla one, which is trained with 64. And so in that sense, our method is better. Okay, so one qualitative result. So here you have an input frame, which is a superposition of two structures, in this case, actin versus mitochondria. Here I zoom in to the section, to the region for which we show the predictions. So this is our input. For this, we have the predictions for the vanilla case, lean LC, deep LC, and the ground truth. So you if you compare the predictions with the ground truth, I hope you see that we are indeed able to segregate the two structures from this input. I will show you more results later. Please allow me to skip the synthetic dataset part because I think I will be out of time if I don't. So yeah, we created a synthetic dataset to show that so this dataset, the property of this dataset was that you cannot perform nicely on this if you do not use the context. And so with this, with our LC, we were able to segregate the input into the two structures. So the summary from this part is that context is useful and variational nature is something which is good to have.
Hi,
Yes,
I'm sorry.
yes, please.
Could you go to the table of numbers?
Yes.
So even in the one with 64 of regular LC, so even there their PSR seems to be a little bit more than for 5 and 2. It's 33.5 versus 33.3. How is that?
It could be, right? Because both of them see the same context. It is just that deep LC is deeper.
No, talking about the previous one. Okay, I see. Okay.
And I also want to say that this is an empirical result, right? So it will depend on so many different things. If you change the learning rate, then maybe the numbers could shift a bit. So yeah. I would not say that.
Yes, no, should
yes.
not be too.
Yes. Okay. Yeah, so yeah, now let's move to the other part. So now we try to solve a harder problem, which is phototoxicity, which in simple terms means that the cell samples, they typically do not like light, which essentially means that microscopists work very less light as possible, which means noisy acquisitions. So now our task was, can we go from this noisy superposition of two structures to the clean estimates? So that is the task. And we train again in a supervised way, but with noisy targets. So we have access to the input, which is a superposition and has noise, and we have the two noisy counterparts, which essentially compose this input. And then we train the network to go from here to here. So this is denoiSplit. We published it this year at ECCV. And this is the overall pipeline. You have the superimposed image, which is noisy, as input. You feed it to our network. We get the two predictions, and then we train this network on the noisy targets. Two things that we changed. One is that we reverted back to the classical K loss formulation, since now we wanted the full denoising property of the VAE-based framework. And the second is that we introduced per channel noise models. Noise models are something that were introduced in our lab for unsupervised denoising. And so let me talk a bit about noise models. So noise model is something which captures the distribution of noise added by a specific microscope setup. So what does this mean? So if you have let's say for a clean value, which I call as signal, it will give you a distribution on the noisy value. So from the microscope, you will typically get these noisy values. And this noise model can give you a mapping from a signal value to the distribution on the noisy values. How do you generate it? There are ways. What you need is essentially you need a paired high SNR and low SNR images. So for every noisy image, if you have a clean version, then you could create such a thing that for every signal, that is every pixel value from here, you can create a distribution over the noisy values. And this we integrated into the, and this is a pictorial representation of this noise model. So yeah, we integrated this into the likelihood computation. So let me explain. You have this input, you feed it to the network, it will give you two predictions. And we assume that, let's say this is clean. And so now you take this pixel value as a signal, you feed it to the noise model, it will give you a distribution over noisy values, and we have access to one noisy value specifically, right? So we have a distribution, we have a value, you could compute the likelihood. And in this way, essentially, you train the network. The benefit of noise model is that different microscope configurations have different amounts of noise. So if you have your noise model specific to your microscope, then we showed in our paper that it indeed gives you an additional benefit in the denoising.
The noise, does it only depend on the pixel in question, or does it?
We are talking about the pixel independent noise that is present in the microscope. We are not considering here the structural noise that is also present, but that is something that is, we are not working on that. Yes.
Sorry.
Yes.
Where you are acquiring your image even on the same microscope, you might have different types of noise.
Typically, you have a Gaussian and Poisson noise. These are the two dominant sources, right? And so both are pixel-wise independent, and so they can be modeled in this way. So you have autofluorescence, you have other kinds of issues, but that is something that we are not tackling here.
This noise model can be viewed as a
Matrix multiplication. I would not say so because here we are not actually doing a multiplication, right? So what we are saying is, let's say your actual signal, God gave you the signal value of 955, but you will not observe this value 955. If you image at that spot, you will get 900 or 910 or 9 something, right? So essentially, so this is what a noise model is giving you. For a signal value, it is giving you a distribution. So there is no matrix multiplication happening here.
So it's like mu plus n 0, sigma square.
And that is yes, closer. Yes.
And the noise is independent of the pixel. Which pixel it is? It's not fixed sigma.
Yes. It is a property of the microscope. It is a property of the microscope configuration. Yes. Okay, so let's now look at some qualitative results, and then we will go back to the other technical aspect of this work. So here again, I show you the same thing. You have the input, you have the two ground truths, you have the prediction by µSplit. So this input is a superposition of these two structures, and the prediction by µSplit, our previous work, and then you have the prediction by the current work, and you have a high SNR ground truth that you just is here for reference. And if you compare the prediction with the high SNR ground truth, I hope you see that we are indeed able to get the structures and we are able to remove the noise. But let's zoom in a bit. So the same thing, you have the input, the ground truth, prediction by µSplit, denoiSplit, and the high SNR. So this is a case where things work, but let's now look at a challenging task where it is not so perfect. So the second one here, let's zoom in a bit. And if you focus on the second channel prediction, so if you compare this image with this image, I hope you see that there are differences. So the network is not able to predict correctly what should be in the second channel. And one could argue that that is because in the input itself, this channel is not present in a dominant way. And so the network really finds it difficult to predict it correctly. And so we thought that it doesn't make sense for a network to give you just single prediction. It should also give you an estimate of uncertainty. It should tell you that here the network is sure, here the network is not sure, and so on, so that you could ignore the bad predictions that the network provides you. And that was relatively easy because we worked with the variational based structures. So they have this notion of stochasticity, which I briefly mentioned earlier, that when you feed this input multiple times to a VAE or to our setup, you will get slightly different predictions. And the reason is that the encoder does not give you an embedding, encoder gives you parameters of a distribution. And every time you sample from a distribution, you get slightly different values. We use this to do sampling, which means that we can get different opinions from the network about what should be the prediction for this specific given input. So you have this noisy image, and you feed it to the network, you get prediction, you feed it again, you will get a different prediction. And here I just take the difference between the two to show that indeed they are different. And the second last, this is the heat map actually. And the second last column is the MMSE, which is the average of 50 predictions. So this tells you the expected prediction by the network for this given input. This was good that now we not only have a single prediction, we have ways to get more predictions. So the people could create a video from this, and they could look at that, okay, this region, there is more variation, and this region, things are more or less stable. And that could be used to avoid working with bad predictions. But we try to quantify this, and so what we did was we tried to do this pixel-wise uncertainty quantification, and the method was straightforward. You feed the input to the network multiple times. Now for every pixel you have, let's say 50 predictions, you take the standard deviation. So this gives you a pixel-wise estimate of uncertainty. That is, yeah, and in case you have the ground truth, then you could also relate uncertainty with error. Because you see, the network could tell you that here I'm very sure, but it could very well be the case that the error is very high. So the uncertainty given out by the network will make sense only if the network is calibrated, in the sense that the uncertainty matches with the error. So if uncertainty is less, the error should be less, and vice versa. And so in this case, we have the ground truth, so we could compute the MMSE error bit for every pixel. And now for every pixel, you have the standard deviation and you have an MMSE error, and we plotted this together. So on the y-axis, you have the error, on the x-axis, you have the variance, and you see a nice linear curve going upwards, which essentially means that when the variance is high, the error is also high. And we did few things here, in the sense that we binned the uncertainties into 30 bins, and then we learned a single scalar for the whole data set, which we multiplied with the RMV so that it aligns as well as it can with the RMSE. So yeah, that was good, but microscopists were not happy. They were not happy because the input was just a simple sum, and the noise was not real, in the sense that we added Gaussian and Poisson noise on. So now we collaborated with different labs across the globe to get different kinds of data. So now we can do 2-channel splitting, we can do 3-channel splitting, we can do 4-channel splitting. And the noise here is actually noise coming from the microscope. The input also is coming from the microscope. We are also now able to work with the Z-stacks. I guess that was the question. So we work with the 3D data as well. So earlier we were just working with normal 2D images. Let me just zoom in. And so every panel you have this input, target, and the prediction. So the white dotted rectangle, which you see here, this is essentially the input. For this, we have the 4 targets. And if you look at the prediction, then you see that the structures are maintained, but the noise is gone. So yeah, that is, and this is another task. I just want to say here that even when the predictions are not perfect, this is still useful because people typically use fluorescence microscopy images to segment things and then track things. So as long as we are able to maintain the morphology that is present for that relevant structure, that is good enough. So even if the prediction is not perfect, as long as the shapes are maintained, that, in my opinion, should have value. So yeah, that is that. Now, in the last part of my talk, I will just talk about the problem that we are currently trying to solve, which is how to create a model which can handle multiple microscope configurations. So let me give you some biological motivation for it. So input generation in our case is sensitive to several factors, in the sense that let's say you have two fluorophores. So if you increase the laser power, then one of the structures will become more dominant. If you, for some reason, the fluorophores are more attached, more densely packed into one of the structures, then that structure will again become more dominant in your superposition. So even when the structures are identical, you can get very different images for the input. And modulo the noise, they are essentially different weighted averages of these two structures. And so now the task that we wanted to solve was, how can we create a single network which can handle all these variations in the input and still give us the two predictions? And for this, we are collaborating from people from Google, integrating their approach, which is INDI. This was a method for supervised image restoration. So there, the idea is that we have two sets of data distributions. One is the input, which will be bad, in this case, which is hazy, and in the second, which is a target distribution, which is where you want to go. So from a hazy image, you would want to go to the clean estimate. And the way it works in INDI is that you pick a number t between 0 and 1, and then you do a weighted average between these two images. So first image is from your target, the other image is from the input. So you create essentially xt. So every time you pick a t, you will mix differently. And in the backward process, you feed this into the network and the t as well. And the task is you train this network to get the clean estimate. So I hope you see that this has very direct relation with the kind of inputs that we had. So our inputs are differently mixed. And from there, we want to get to the clean estimates of the individual structures. So the inductive bias of this INDI network matches perfectly with the inputs, the way inputs are generated in microscopy. And so since we have two targets, we now have two INDIs, one for each channel. And we do the same process. We mix. So the two distributions that we have are the two target distributions, and we mix them with this random value between 0 and 1, and then the network is trained to predict one of the channels. One part is missing, which is that for prediction, we also need an estimate of t. But from microscope, you will just get a superimposed image. And so we trained another network, which is a regressor network, which takes as input this superposition, and it gives you an estimate of t. And then we feed both of them together to the network, and this gives us the prediction. This is work in progress, so I will show you two qualitative results here. So here you have, again, actin versus mitochondria task. You have two rows because you have two INDIs for the two tasks. This is the input. In this case, both are identical. The two INDIs go from this input to the relevant structure that they were trained on. And if you compare the target with the prediction, I hope that you see that there is a very close resemblance between the prediction and the target. But this is not, let's say, super interesting. What is interesting is that the same network can also remove the faint structures that will happen if one of the structures is dominant and other is not dominant. So this case is important in microscopy, and it goes by the name of bleed through. So here you have one dominant structure and the other bleeds into this. So you see some remnants of this structure present here, and the network is still able to remove, the same network is able to remove these faint structures, as you can see in the prediction. So the same network can handle different kinds of differently mixed inputs.
So
Yes.
So what if your intensity of the structures is not uniform? In that case, what if the specs that you see in the bottom channel, they're actually because the structure is organized as the intensity is lower in some parts and higher in others.
Right, that can happen.
The intensity of your sample also lead to the erasing of that particular image. Is there an artifact to the system?
So, okay, so it depends on the kind of training data you have. Let's say in your training data, you have such a thing, then the network will learn to handle this. That there will be some bright spots and some dim spots. The network can handle that. But of course, if you have limited amount of data, then what you're saying will happen. Because then the network will assume that the intensities will be between these ranges, and then it will have issues. Yes. So my, let's say, simple advice would be gather more data to train. And if that is, I cannot put a number to it, but we work with, let's say, 2000 cross 2000 size frames of 100, let's say 10 stacks, let's say 10 stacks. And what is important is that they should be different, because sometimes what happens is that you have very similar structures. Then it doesn't matter if you image multiple things which are very similar. So yeah, one more work, but I will not present here. So this is that while we were doing all this, we figured out that structural similarity index measure, which is a very popular measure for images, natural images, does not work well for microscopy data. And we figured out the limitations, and we provided a way of solving those limitations. We published it at Bioimage Computing Workshop, ECCB, this year. This brings me, in the last 5 minutes, I would like to talk about the future that I would want to work on, and that is on image translation in biological and biomedical data with a focus on uncertainty estimation and calibration. So we already saw image splitting, which hopes to empower how multicolor imaging is done. The same setup could also be used to remove structural noise for some cases. If you are able to model structure as a separate channel, then you have one channel where you have the clean data, you have the other channel, which is the structural noise, and the input that you typically have will be a superposition of these two things. And so you could also solve structural noise with this image translation kind of network. And we solved one of them, so which is we try to remove the punctas, which you see as bright dots from the image. And if you compare the inputs and the prediction, I hope you see that the punctas are gone. I do not claim that every kind of structural noise can be removed with an image translation network, but if I spend enough time, then I hope that I should be able to remove different kinds of structural noise with this approach. The other popular, let's say, emerging field is this field of virtual staining, where people go from transmitted light image to fluorescence image. And what do I mean is that fluorescence microscopy is nice in the sense that it is specific. If you want to image nucleus, it will give you nucleus. But with transmitted light, you get everything. So why people use transmitted light? They use it because you cannot use fluorescence microscopy for longer durations. It is too toxic on the cells. And so this virtual staining tries to get best from both worlds, in the sense that the idea is to image using transmitted light image and then convert it with deep learning to some specific fluorescence image which you care about. And it has applications in histopathology, it has applications in other cases as well. So, and it also has applications in segmentation. For example, if you want to track nucleus from this image, you would not be able to do so because it is difficult to even train a network to segment nucleus. But if you were to convert this image to a fluorescent image for nucleus, then from here to here it is trivial. You just have to apply a threshold and you can segment things. The problem with all these things that I showed you, that they all are currently giving you a point estimate. So they don't provide multiple predictions. They don't provide any way to give uncertainty estimations from the network. And there is not much notion of calibration. And so this is something that I would want to work towards. I showed you one calibration plot, but the limitation of this thing is that when the data is out of distribution, in the sense that you use a different microscope configuration to generate the data, then this thing will not hold. And so to do a proper job at this, we need to also tackle the case of out of distribution, where the images will come from a slightly different data distribution, and then how do you handle it? So in general, uncertainty estimation has multiple use cases. For example, you can use it to ask this question that, can you give me multiple predictions for this input? Or you can ask this, that you have a pre-trained model, you have an input. Is this input suitable for this model? In the sense that, is this input out of distribution with respect to the data on which this model was trained? And you could also ask this question, that how much training data you need to train a given model, because this is important in biological and biomedical domains, because data acquisition is sometimes costly. Now, The first one could be answered by what people classically term as aleatoric uncertainty, and the next two questions can be answered with epistemic uncertainty. Aleatoric is something which is an uncertainty which is inherent in the data. For example, if I ask this question that train a network to predict positive summands for a number, right? So if the number is 20, then 1 and 19 is a good answer because 1 plus 19 is 20. But the same 4 and 16 is also a good answer because that also sums to 20. So there are problems where a single input can have multiple solutions. And for such problems, it does not make sense to give you a point estimate. You need a way to do posterior sampling, which means that you need to have different predictions coming out from the network. The second is epistemic. This is something that could be reduced. So if you have a better model or if you have more data, then this component of uncertainty can be reduced. I have started with, let's say, doing preliminary research on this. I tried to implement Monte Carlo dropout into our setup. This is essentially you use the dropout, but you use it during inference time. And so essentially you are predicting from many different networks. And I try to answer this question that how much training data is needed. I will just tell you the intuition. The intuition is that if the training data is less, then the uncertainty will be more. And so if you just quantify this plot, then it might give you a handle on how much training data you would need for your task. For the question that is the input out of distribution, you can simply plot the distribution of the uncertainties from your training data. And if you see a difference in that distribution, when you plot the same thing on the data that you currently have, you can let's say answer this question that is the input output distribution. I need to do more work on this, and I will be going to this ENS lab in France under with Julien Taquel. I'll do an internship with a focus on creating better uncertainty estimates on the task that I'm working on. And with that, I would like to thank Florian, you, my PI, who is here, half, and all my UGLA members and bioimage analysis members, and all the excellent collaborators who have given us the data and technical insight on this task, and all of you for your patient listening. Thank you.
Questions?
So you said that you developed the denoiSplit to help you with both low signal sampling,
Right.
low SNR sampling.
Right.
So in that case, your input indicated both a high SNR and a low SNR image of the same image, is it?
So in the denoiSplit, I mentioned that I work with synthetic noise. So therefore I had access to the high SNR with which I could compare. So that was just for comparison. We did not use the high SNR in the network.
Okay, so the training data set is still.
It's just noisy. It's just noisy data. Just noisy data. Yes.
So it's not necessary to have high and low SNR.
No. So for noise model generation, you need it, but there are ways around it. For example, you could use, you could sample the same structure multiple times and then average it. That will give you a high SNR estimate, right? Or what you could do is you could use some unsupervised denoising on the low SNR. That will again give you a high SNR estimate.
More questions? So let's thank you again.